So I'm left with nothing. I appreciate that. Yep, me. I ate that whole pizza. So fatty. How many pizzas did you get?
9. No. Oh yeah, you're not.
Wait, does yours have sausage?
No, it's chicken.
Is it barbecue chicken?
Yeah, there's barbecue chicken. Wait, go get it.
You're saving me a piece of that.
You want to bite? It's from like. No, but it's like. Oh, do you have to get it from the kitchen?
Here. Just rip off a piece and put it on.
Do you like the crust or do you like the?
Attention, Kaiser Whitney and Cody Parker. Please report to S6, Mr. Kukowski. Once again, Kaiser Whitney and Cody Parker.
Okay, now I have.
Report to room
Paper towels.
S6. Thank you.
All right, you guys. Please get off your phones. Open up a textbook. Be ready to listen and answer questions. So we're going to talk about plasma membrane first, and then we'll talk about structure. So the plasma membrane is made out of what main molecule? You guys, I would really like you to be quiet. Yes, its main component is a phospholipid. And it is in a bilayer. Okay, so just to review, when we are drawing it, we have this region, which is the head, and the head is hydrophilic. Hydrophilic, which means what? Likes water.
You say phospholipid bilayer. Is there any other kind of phospholipid layer?
Well, a phospholipid is just this. The way that all of the plasma membranes are set up is for it to be a bilayer. But we could be talking about just one phospholipid. All right, this region is the tail, and it is hydrophobic. Other terms that we use other than hydrophobic, we also say that it is
Polar?
the tail is nonpolar, the head is polar. Okay. All right. The fact that it has these two regions. The name for having these two regions is amphipathic. Okay.
Just meaning that it has the hydrophilic head.
And the hydrophobic tails. All right, so we good with that? Main component of it is the phospholipid. It's in a bilayer and it has those sections. All right, we say that it has a fluid mosaic model. So there's the fluid mosaic model of the plasma membrane. We're going to talk about the mosaic part first, just because I think it's easier. Mosaic means it's what? Made of different things. And instead of things, we say substances. So what are the things that are in, other than, we have the phospholipids.
Proteins, cholesterol.
What else is in the membrane? We have proteins. What else? Other lipids. What other type of lipid is in there? Cholesterol. What else? There's another type of lipid as well. Those one green things. Long green things. What are those long green things? We have glycoproteins. We also have Glycolipids. So the glyco means what? Carbohydrate. And a lot of times these are going to be the what? The identifying tag things, also known as receptors. Okay. The carbohydrate, glycoprotein, if you have the book open and you're looking at the picture of the plasma membrane, they are the Y-looking things that stick out of the plasma membrane. So if that carbohydrate is attached to a protein, then it is called a glycoprotein. If it is attached to the lipid part of it, then it is called a glycolipid. Okay, but either way, they're both a carbohydrate. It's just what is attached to that is different. Okay, so this is why it is a mosaic, because it is made up of many different substances together. Okay, and proteins, we do have two different types of proteins. What are the two different types of proteins?
Channel
No.
areas?
Peripheral and periphery. I can't spell. Is it a so we have integral and it is a pH. Peripheral. Peripheral. All right, so peripheral does what? On the what?
Sorry.
On the outside or on the inside? Basically, these ones do not go through the entire bilayer, right? So they could be partway in the membrane towards more the cytoplasm side, or partway in the membrane more towards the cytosol, or intermembrane fluid area. I'll draw a picture in a sec. But they do not go all the way through the membrane. Whereas the integrals go all the way through. All right, so if we're drawing this, so if we have this protein that is going all the way through the lipid bilayer, then this is going to be the integral. Let's say that this is the cytosol and this is the interstitial. What do they call it? The
Intermembrane space.
No, it's not intermembrane. All right. So these proteins can be more towards the outside of the cell or more towards the inside of the cell. Okay. So it doesn't matter. They're just not all the way through. Okay. And those would be the peripheral. Okay. Are we good? All right, so the fluid parts. All right, so fluidity means the ability to do what? To do what? To move. So if you were to think of the cell being a 3D shape, kind of like this, right? On the outside, if you were just viewing it from the outside, we would see a whole bunch of the heads, right, of the phospholipids. And we wouldn't really see the tails if we're just looking at it from the outside. So it would look very similar to if you were to look at people walking in New York or something, right? And all we are seeing is their little heads, right?
buildings with those proteins?
Sure, the buildings can be the proteins, okay? We're looking at a bird's eye as if we were in a plane above them, hovering, okay? We're in a helicopter hovering above the busy streets of New York, and it's packed with people, right? And all we can see is their heads. Their movement are the same way that phospholipids can move, right? So if I need to get over there and I have to go through people, then my little head, I would just see it moving, right? So phospholipids can move, okay? And we call that movement, if we're just going from here to there, what type of movement? What direction is that? Lateral. So phospholipids can move laterally very easy.
And the super performers are the ones that like flip upside down.
There you go. Complete analogy. All right, so they can move laterally really easy, okay? They do not move horizontally or flip sides of the phospholipid bilayer very easily. Why is that? Because that polar head does not go through that non-polar region of the lipid bilayer very well at all, okay? So can they flip? Yes, not nearly as often as they're just moving laterally, okay? So they can move laterally really, really easy. Okay, can proteins move as easy as the phospholipids? Not as easy, but can proteins move if they need to? Yes.
Just please start working. Just go. All of it. Just all right. Just. I'll pick it up. I'll pick it up. Squares don't go in circles.
All right. So proteins can move as well, but not nearly as easily. Okay. And we do want to have this movement. Okay. The fact that the plasma membrane can move is significant, especially when we're talking later on about receptor-mediated endocytosis, because we need those glycoproteins to all group together, and then we can endocytose in that position. So the fact that they can move is very important, okay? There are things that will affect the fluidity of the plasma membrane.
Cholesterol.
Temperature is the number one, okay?
Saturated and unsaturated.
Saturated and unsaturated tails are going to be another thing that is going to affect it. And then the last thing, pH can. What else? But pH is just it's kind of similar to temperature. And then also cholesterol, right? So if we're looking at the things that affect it just individually. So the first one we'll talk about is temperature. Okay, so these can affect fluidity. All right, so temperature will affect fluidity in what way? Okay, if it's warmer, they are able to move more. Okay, so what if it's hot? It will denature the protein. So it's good to be warm, but not good to be hot. Okay, so if the so in general, as the temperature increases, fluidity will increase until it reaches its max ability to withstand that temperature. Then it will denature. Okay. So if we're looking at a trend in general, as temperature increases, fluidity increases. In general, until it's too hot. Okay, then it will denature. And what is it denaturing? Mainly the proteins, right? The proteins are the things that are really sensitive to temperature. Okay. it goes with saying that if a warm temperature allows them to move, then a colder temperature hinders them from moving. Okay, so in general, as temperature is increasing, we will increase fluidity. So the colder the temperature, the less movement we're going to have. Okay, so it kind of looks like, if we were to look, fluidity would increase until it reached its maximum, and then it would, when it denatures, it would decrease.
For the lateral and flip-flopping
Uh-huh.
stuff of the liquid bilayer, is that for room temperature?
If you want to think about it that way, yeah. I mean,
Because if it would be slower, then it wouldn't be that, or faster.
correct. It would, well, the ease with which it could still move, it still has the ability to. It might not in colder temperatures. So until we put a specific situation on it, I want you to think best scenario, can it? Unless you're given something else, okay? All right, so that's temperature. Along with temperature is pH. And this is how pH would affect the proteins, right? So really, pH is going to affect the proteins more than anything else, okay? And we know that usually proteins have an optimum pH. So if the pH is too low or too high from the optimum, then what will happen?
Nature,
Then the proteins are going to denature, and then what effect would that have on fluidity?
less fluid.
It would decrease the fluidity. Okay.
Would it be more fluid though if it has more space? If it
No, when the proteins break down, it's not. It messes up that part of the plasma membrane.
Oh, is that why things get harder when you burn them?
When you melt them,
Yeah.
well, when you melt them, you change their configuration, and then usually what's left over is after it's cooled from that hot temperature. You're left with that burntness, right? So a lot of times when you heat something up, you're changing the composition. You're oxidizing it. You're doing things to it that change.
So with yeast,
What?
what's happening with yeast?
Well, organisms are different than compounds, right? Okay, so in general, those are the two. Okay, the next one is the presence of cholesterol.
Steve Gunning, please call 3567. Mr. Gunning, please call
So
3567.
in general, if we are adding cholesterol to the plasma membrane, what is that going to do?
Buffers the temperatures.
No, we're not talking about the buffering of the temperature yet. We're just asking about having cholesterol be part of the plasma membrane.
It keeps everything
As
like
far
the
as
same.
fluidity,
As far as fluidity, when we add cholesterol to the plasma membrane, what does it do to fluidity?
It prevents any change, so like,
No,
oh.
makes
it
warmer,
decreases fluidity.
less-
Okay? What I want you to think of is if this is our plasma membrane, right? And we have a certain amount of space, and then we start adding cholesterol into it, we're packing things in, right? We're not getting rid of anything, we're just adding something new. So now that's like adding more people to the group, and it's harder to maneuver, right? So we're less fluid. So adding cholesterol will decrease fluidity. Okay? There's just one more thing in the way. All right? And then the last thing that is going to affect, and we're going to come back to cholesterol, but I want you to know cholesterol by itself. Okay? So just if cholesterol is there versus if cholesterol is not there. Okay? Temperature not being a factor. Okay? Room temperature. Temperature is fine. If we have cholesterol, more of it, the more cholesterol we have, the less fluid the plasma membrane will be. Okay? And then the last thing is whether we have saturated or unsaturated tails. Okay? So saturated have what shape? Unsaturated are kinked. Why are they kinked? No, not because of hydrogen bonds. Carbon bonds, what about the bond? It's a double bond, okay? So saturated, it's completely filled with hydrogen bonds. There are no double bonds, so it's straight. Unsaturated, we have a double bond between the carbons, so we get a kink. What does the kink allow there to be? More room, right? It's like sticking its leg out and saying, give me some space. Okay, the straight ones get packed in. So which one is going to allow for more movement? The kinked, right? So if we have a more saturated, then we're going to have less fluidity. Right? If we have more unsaturated, then we'll have an increased fluidity. Are you just chilling here or do you need something?
I'm waiting to send you some candy.
I'm not gonna buy candy grams. Go steal some of my money from Savannah Avery.
My kind of freeze. I'm gonna sit right here.
Just had a $7 for me.
I want to listen. Just be patient.
Sam,
I'm not distracting you.
but you are.
All right. Cholesterol is also called a buffer. Okay. Temperature buffer. Now, this is what I do not want you to say because a lot of people said this. Cholesterol does not increase or decrease the temperature. Okay? It cannot do that. So the temperature is going to be whatever the temperature is going to be. But cholesterol can help the plasma membrane maintain its fluidity because a buffer wants to resist change, right? So if there is a change, then it will try to counteract that in some way. Okay, in this case, it would be by either increasing the cholesterol in the plasma membrane or decreasing the cholesterol in the plasma membrane. Okay, but cholesterol does
Just a reminder
not
to all
change
students,
the
tomorrow is
temperature.
the last day to buy holiday grams for that special someone. Find somebody in the ASB, each holiday gram is $1. Again, tomorrow is the last day to purchase holiday grams for that certain someone.
All right. So if temperature becomes an issue, then cholesterol can act as a temperature buffer in that it tries to keep the fluidity the same. It tries to keep the fluidity constant for as long as it can. Okay? So if the temperature increases and it's getting too fluid, right? So if we have an increased temperature and it's moving more than maybe the plasma membrane should, then we can increase or decrease our cholesterol to maintain it. Okay, if we increase temperature, what does that do to fluidity? It increases fluidity, right? But if we want to counterbalance this, then what we need the cholesterol to do is to decrease fluidity, right? In the same amount that the temperature increased it. So how can cholesterol decrease the fluidity? Add more cholesterol, right?
Cells create it or just like fluid?
Well, it's a lipid, so it could be created by the cell. So what's going to happen is we're going to add cholesterol, right? And if we add more cholesterol, then that by itself would decrease fluidity. Temperature will increase fluidity. So if we add cholesterol, then we're keeping it constant.
Did you know you talk about how your cholesterol is too high? Are you supposed to take it out so it makes
Make your computer easier to use.
Sam,
I'm not trying
back away from my personal area. That's a little bit different. This type of, that's like the cholesterol that's just circulating in your blood, and we don't want too much of the bad stuff because it clogs our arteries and gives us heart disease. Okay. All right, let's say that the temperature decreases. Okay, the temperature is too low. What is that going to do to fluidity?
I don't know for you, but I can come to your class. This is awesome.
Sam.
No, keep going.
All right, so if we decrease the temperature, that's going to decrease fluidity. The way that cholesterol can buffer this is to do what?
Take more out.
take cholesterol out.
When, after school? Okay. You can always just pay me tomorrow. We're selling them tomorrow too.
All right, and now if we take out the cholesterol, it will keep the fluidity constant.
I'll just put you on my list.
Okay.
I can find
I
the
didn't
box.
get to transport, so I don't know how you guys want to try to do that. If you want to just ask me in class, but transport would be diffusion, osmosis, facilitated diffusion, and bulk. So I know AP kids, we have class time, so if you want to ask me about those because you're unsure, then we can.
Honors we have.
Yeah.
Yeah, we have lots of
The what?
Oh my god, piece of pizza, yes.
I figure if I stay here long enough,